Nagaland University pra Berhampur University aro Savitha Medical College logode milli gina study ekta kuriya de researchers khan paishi ki Konyak manukan traditional herbal chulai thaka khan cancer bhal kuribole potential ase. Researchers khan molecular simulation nishna advanced technique khan chulai gina cancer laga khilaf kuriya compounds khan identify kurishi aro itu khan dawai khan nishna kam kuriye kintu toxicity komti thaka paishi.
Drugs for cancer. That compounds are called as anti-cancer agents for the colon cancer. So in the anti-cancer plants, we have chosen 5 different plants. In these 5 different plants, these species are having good anti-colon cancer property, so that we, from the tribal healer knowledge, that is they are the informants now. From the informants information, we have identified the 5 species where the 5 species having the potent anti-cancer activity. So then finally we discovered 3 important compounds. One is the colistin, and another one is the progenon, and another one is afitinib. So these 3 are the 3 important compounds which it is playing as a important anti-cancer agents from the formulation which we have made from the 5 important plants in which the tribal healers gave. So we have proved that the compounds which we identified from the formulation against the colon cancer cell line, we have grown the colon cancer cells, that is the HT29, in the laboratory. Then we treated that cancer cells with the different extracts of the identified plants. So in each plants we find out the potent anti-cancer property from the 5 different plants. Then we have formulated the 5 different plants. That formulation is in the form of small powder. That powder we can also intake by decoction method. So just 1 gram of powder you can add in 2 glasses of water and then boil well. After it was condensed into 1 glass of water which contains the decoction, that decoction if you intake, it is worked well against the colon cancer. So that this formulation is proven in laboratory stage. So further process is needed to tested in animal as well as human. But the same formulation in which the healers also, they are administering to the patients, those who are trusting the healers, they are treated the patients with these same plant extracts. Now from this study we are scientifically validated the extracts of the tribal healers. We evaluated the formulations of Konyak tribes, what they are administrating to the patients, cancer patients. So we have the one of the rotary evaporator. We have one of the important instrument. So this instrument is used to extract the extracts from the plant's crude plant leaf powder or root powder, so like that. And then we have a spectrophotometer to identify the compounds which is present in the plant extracts. And we have the centrifugation instrument. Centrifugation is basically used to separate the high molecular compounds and low molecular compounds. And then we have other setups which is required for the extraction, like Soxhlet extraction. The Soxhlet extractions are used for extracting using different solvents like polar and non-polar solvents.
So what we basically do is like we interact with the traditional healers, and then after that we collect a medicinal plant. So that that medicinal plant extract, we do the preliminary investigations such as the antioxidant potential, and the anti-cancer potentials on the cell lines like HT29. So with that, we further proceed with the other spectroscopic and chromatographic techniques such as GCMS analysis. From there, we identify the compounds, and those compounds, what we do is we use certain softwares like talking softwares and the molecular dynamics. Like we do analysis on that, and then we also check the toxicity and the trackability of the compounds as well, and the pharmacokinetic activity of the compound. In our recent research, what we have found is there were certain compounds which have the capability and then which have the potential, like comparing to the standard anti-cancer compounds, like which have been used for the treatment of cancers. But these are all preliminary investigations, and for that, we have to do further in vivo analysis, so which will confirm how efficient the drugs will be.